Hello, Sreenath sir. Welcome to the National Webinar Series, and we are going to start in another 5 minutes. So it's still 10:50. So exactly at 11, we are going to start. Hello, Sreenath sir. Is audible? Is hello? Hello, Sreenath sir. Mute ಆಗಿದೆ ಒಂದು. Hello, Sri. Hello, Sreenath sir. Can you hear me? Hello? Hello, Sreenath sir. Can you hear me?
Hello.
Hello? Oh, yeah, Sreenath sir. Yeah, now I can hear you. I think it was unmuted. Can you able to hear me?
Yes, sir. I can hear you.
Yeah, fine. So you just keep that. We're going to start in another 2 minutes. So you can be on.
Sure. Thank you.
Yeah. Hello, Shankar sir. Can you hear me? Hello?
Hello.
Yeah, Shankar sir, is it I'm audible?
Hello.
Yeah, I can hear you. Thank you.
Dr. Pramod, can you hear my voice?
Yeah, yes.
Okay.
Hello everyone. Very good morning and welcome you all to the National Webinar Series of the session scheduled at day 7. So today on 21st June 2020, we have one valedictory session and then the last speaker, the valedictory keynote speaker, which is scheduled at 11 to 12 is Dr. Sreenath Kundami, Head Cell and Molecular Biology, Laila Nutraceuticals, R&D Center, Vijayawada. And Dr. Sreenath is going to speak on cell-based therapeutics, focus on cancer immunotherapy. And this will be followed by the valedictory sections at 12 to 1. And I now request my friend and our colleague, Dr. J. Shankar, Associate Professor, Department of Food Technology, to introduce the speaker of the session, Dr. Sreenath Kundami. Over to you, Dr. J. Shankar. Shankar, sir, please.
Thank you, Dr. Pramod. Good morning, dear participants. Welcome to the day 7 session of National Webinar Series 2020 on infection and immunity, biochemical approaches to therapies. The today's speaker is Dr. Sreenath Kundami. Presently is the Head Cell and Molecular Biology, Laila Nutraceuticals Research and Development Center, Vijayawada, Andhra Pradesh. So just with a brief introduction about his education details. So he did his BSc Ed from Regional Institute of Education, Mysore, and he obtained his MSc degree from Department of Biotechnology, University of Mysore, PhD in biochemistry from CFTRI under the able guidance of Dr. Y.P. Venkatesh. Later he moved to USA for his postdoctoral studies, where he joined the clinical immunology of trauma and brain patients, immunological role, alcoholism at a Department of Surgery and Research, University of Rochester Medical Center of Rochester, New York, USA. And next he moved to University of South Carolina, Charleston, South Carolina, USA, at the Department of Microbiology and Immunology to pursue his second postdoctoral studies. His professional experience: Assistant Manager from 2013 to 17, Department of Cell and Molecular Biology, Laila Nutraceuticals Research and Development Center, Vijayawada, Andhra Pradesh. Head presently is holding the position Head Cell and Molecular Biology, Laila Nutraceuticals Research and Development Center, Vijayawada, Andhra Pradesh. Member, Institutional Animal Ethics Committee, Laila Complex, Vijayawada. So member, Preclinical Efficacy Committee, Laila Nutraceuticals, R&D Center, Vijayawada. Member, Clinical Protocol Committee, Laila Nutraceuticals, R&D Center, Vijayawada. There's a vast experience in the field of immunology and immunotherapy, in vitro, preclinical efficacy and clinical studies, adaptive T-cell immunotherapeutics in preclinical models, preclinical efficacy models of osteoarthritis, obesity, muscle endurance, and brain health. Project proposals, management and completion, collaborative multidiscipline projects between Laila Nutraceuticals and other institutions. So to mention few of his publications, he has published over more than 12 research articles in peer-reviewed international journals, to mention a few journals such as Clinical Cancer Research, Journal of Immunology, Bioconjugate Chemistry, Analytical and Bioanalytical Chemistry. So he has published 4 book chapters and he has 1 patent to its credit. To highlight some of his achievements, 2 Young Scientist Awards from Indian College of Allergy, Asthma, Applied Immunology, Indian Academy of Allergy during his PhD, 2 Travel Awards from Society of Leukocyte Biology, USA, Federation of Clinical Immunology Society, USA during his postdoctoral studies. CSIR NET Fellowship from CSIR for JRF and SRF. Best Research Paper Presentation Award, Federation of Clinical and Immunological Societies, 2009 Conference, San Francisco, USA. Teaching certificate from Medical University of South Carolina, Charleston, South Carolina, USA. And is a member of some few professional affiliation, past and present, to mention a few: Society of Immunotherapy of Cancer, USA, Federation of Clinical Immunology Society, USA, Society of Leukocyte Biology, USA, Society of Biological Chemists of India, Mysore chapter, Mysore. With this brief introduction, I'll invite Dr. Sreenath Kundami to take over to present his talk. Over to you, Dr. Sreenath.
Thank you, Dr. Shankar. Am I audible, Dr. Shankar and Pramod?
Yeah, you're audible. So voice is clear.
Okay. Thank you, Dr. Shankar, for those kind words. And Dr. Pramod, I would like to thank Dr. Pramod and the organization committee for giving me an opportunity to present my experience at National Webinar Series 2020 held at Davangere University. So to start with, I focus my topic on cell-based therapeutics and especially more focus in the area of cancer immunotherapy. Next slide, please. So if you let us look at the growth of pharmaceutical industry. So most of pharmaceutical industry before 1980 was on small molecular based drug discovery. So in the early 1980s, with the advent of biologics like recombinant proteins, soluble receptors, antibody based drugs, so then the treatment shifted and paved a way for broad range of new targets and functional capabilities, also for various disease applications. Now, as of today and early 2017 onwards, biomedical science stands poised at the threshold of another pharmaceutical frontier, which is cell-based therapeutics. Next slide, please. So let us look at the advantages of therapeutic cells as a therapeutic agent. So there are main advantages to talk about. Firstly, cells can perform naturally therapeutic tasks. For example, macrophages can engulf pathogens and recruit adaptive immune cells at the site of infection. This is natural. And HSCs, like hematopoietic stem cells, can have myeloid and lymphoid lineages and have immunological activities. Chondrocytes, for example, can produce extracellular matrix. And pancreatic beta cells can sense glucose and produce insulin. These are all natural activities for cells, for example. Like gut bacteria also can convert indigestible fibers into SCFAs, which is the short chain fatty acids, and that can fuel intestinal epithelial cells. And also, if you look at cells, their behavior is very selective. Like they can sense their environment and respond accordingly to the signal they receive. So they can have kind of on and off switches. If the signal is go, then they will respond. If the signal is not there, then they will not respond. So cells also could be a special delivery agents, in the sense they can recognize, actively migrate towards a specific signal and then perform their activity, so that you will have less off-target effects, like in the case of most of the side effects due to small molecular drugs. For example, rosiglitazone, we know that it induces PPAR gamma activation. Instead of that, if I can engineer a cell to produce ligand, PPAR ligand, which specifically goes and bind to PPAR gamma only, as rosiglitazone has other side effects, as we know. So cells also can handle genetic variability. So if we can engineer a cell to produce an unnatural task also, like a modifying a T cell to sense glucose, which would be a provocative alternative, but of course we have to keep in mind that the risks associated and how to handle the risks. Next slide, please. So let us look at how evolution happened of cellular therapies. So with the advent and advancement of systems biology and synthetic biology, so which paved the way for cellular engineering basically. So where we can see that engineered cells can control their response, can activate depending on the signal, and proliferate and execute their tasks. And also you can program a cell death also, like engineering a particular enzyme like thymidine kinase. You can redirect migration also if required, so that they respond to the signal and then work. And you can also engineer in such a way that they can make their own decisions depending on the situation they are put in. Also, they can have a multi-agent cooperation also, like they can have sense signals from to and from different cells and respond accordingly depending on the situation. Also, they can be engineered to produce a specific small molecules or biologics like cytokines to have a particular targeted effect. So this leads to the fourth pillar or the fourth pillar of therapeutics, which is called cell-based therapeutics. Next slide. Next slide, please. So let us look now. This is the age for apps. So let us look what apps we have on our hand for cellular therapy. So if you look at the top left corner, this is the you can engineer a cell to sense a particular tumor antigen and specifically infiltrate that. You can have a transplanted microbes which can sense the inflammatory signals from mucosal epithelium from a gut and then generate anti-inflammatory secretions so that you can reduce the inflammation. This would be very helpful in the case of IBD, inflammatory bowel disease, and Crohn's disease. Thirdly, you can have a coordination between two engineered cells, like one engineered cells and transplanted microbiota, so that they can signal the dietary molecules and make engineered cells to produce GLP-1 specifically, which can induce satiety. Fourthly, you can collect, it is well known that now finally terminally differentiated cells from any tissue or any organ can be cultured, and then you can induce pluripotency through some reagents and have induced PSCs, pluripotent stem cells, and with that stem cells you can direct whatever population you want to get and then re-inject into the human cells. Next slide. So let us focus on tumor immunotherapy for this talk. So what is adaptive T cell immunotherapy of cancer? So if you look at the history, so cancer and immunity, so around 50 to 60 years ago, the syngeneic mice, when they were transplanted with tumor cells, they actually showed rejection. So that is kind of a starting point, takeoff point for the immunity and cancer. Now, around 25 years ago or 30 years ago, tumor antigens were identified and can be recognized by T cells and specifically can target that particular tumor. And if you see the three phases of immune surveillance, so there are three phases, which is called the 3 E's: elimination, equilibration, and escape. First, elimination is when there is a proper immune surveillance, so your tumor cells will be identified. The important with tumor cells versus an infection is because tumor cells are also self-antigens, right? So they produce self-antigens. So the immune cells should be discriminatory to select only tumor antigens and then try to act on them. So this will be elimination process. Next phase is equilibration process, where the number of tumor cells also increase as they grow very fast, and then they try to take over immune cells. So where immune cells and tumor cells reside together, which is called equilibrium phase. Once the stage is out of control, where the tumor takes over the immune cells, then there will be an escape, and solid tumors can form malignancies and enter everywhere. Next slide, please. So if you look at the T cell activation, let us focus on T cell activation, because these are the cells we will be using further to use as a therapeutic agents. So first, on the left, we have a dendritic cell, which is DC, which is a professional antigen presenting cells, which presents antigens to either CD4 or CD8 T cells. The first signal required is a TCR engagement between these two. TCR engagement from the point of T cell and either MHC1 or MHC2 based on the type of T cell. If it is CD8, MHC1, and if it is CD4, MHC2. Once this signal is up, then there are two signals that should take place. So it can be either one. Signal 2 can be CD28 or CD40 ligand on T cells, which is a go signal, which is a co-stimulatory. The second signal, I can call 2B signal, is a co-inhibitory, where it expresses PD1 or CTLA4. So it damps the immune response, which is called co-inhibitory. And the third and final signal is very important, which we require, is cytokine priming. So where once we have the activated DC presenting the antigens to the T cells, it also produces cytokines that can act on T cells and make them proliferative into large numbers, so that they can reach the target and attack it. Next slide, please. So in 2018, if you can recall, so these two gentlemen, Dr. James Allison and Dr. Tasuku Honjo, received Nobel Prize for their groundbreaking discovery of two important inhibitory molecules expressed by T cells. One is CTLA4, cytotoxic T lymphocyte antigen 4, and the second one is programmed death cell 1, so that is PD1. So these two inhibitory molecules are very important, because whenever tumor wants to escape, it engages the T cells, whatever is infiltrating into the tumor, and make the T cell to express these inhibitory receptors, so that your immune response actually should go to activation. So rather going to activation, it goes to a regulation mode, where it suppresses the immune response by expressing these two inhibitory molecules. So targeting these two molecules across different cancers would be a very important target activity that we can focus and then kill the cancer. And there are several number of clinical studies have been performed in various tumors, from lung tumor to any other tumors, lung tumor or liver, any other tumor. Next slide, please. So if you look at this cartoon, so this is the basic discovery what they did. So if you look at top panel, which is labeled as yellow, CTLA4 or PD1. So let us assume there is a cancer cell and there is a APC and T cell engagement is going on. Now you have, we can bring in the analogy of car brakes. So left side I am putting a car handbrake, and right side there is a leg brake, right? So cancer cell induces these APCs to express T cells inhibitory receptors. So where CTLA4 and PD1 goes up on T cells, and then it damps the immune response or infiltrating T cells into the tumor site. Now what happens when you develop an antibody that is anti-CTLA4, the down panel, anti-CTLA4 and anti-PD1, which specifically targets these receptors, CTLA4 and PD1. So it blocks the activity of these two, PD1 and CTLA4, so that they cannot engage with their ligands. Hence, these T cells will be suppressed, and then your APC can present these tumor antigens to remaining other T cells, so that they can enter and kill. And the brakes are off at this place, and they can come into the tumor site and kill. So this is because these receptors express across the different cancers, and then most T cells express this. So that's why when they discovered this was taken huge plus across the cancers, irrespective of what type of cancer, because these inhibitory receptors are expressed in T cells anyway. So if I put brakes or release brakes, so that I can have better immune response to the tumor. Next slide, please. So taking at the same time, around 1980s or 1985s, at the same time, people also thought, okay, now we are working on one antibody. Why don't we collect some cells, engineer them, and put it back? Can we have a curable disease, or at least we can have a regression into tumor? So they looked. So in the initial phases, they looked, okay, where to look at, right? This is a first question. So where they looked was the tumor itself, because they collected the tumor. So if there are T cells reside in tumor, that means they have come for some purpose. So they collected the tumor and excised and make the tumor into different fragments, and they cultured with IL-2, and they expanded into multiple specific clones of different antigen specificity of that particular tumor. And then once they have antigen specific, major antigen specific tumors in large populations, they expanded into much more population. And then after they expanded into multiple numbers in vitro or ex vivo, I can call, in the lab, then they infused into the patients which has been who has been conditioned. What do I mean by conditioned? Because they have to slightly lymphodeplete the patient before infusing these cells, because these are immune cells, then they will have a reaction. So that's why, to avoid that, they will just have a small lymphodepletion regimen, and then they will inject these. So this is the first initial phases where they looked at. Next slide. So later, with the advent and the advancement of gene technologies, so they modified T cells. So you can have number of now tools to modify a particular T cell or any other cell, to modify to have a specific activity. So in this, three important components are important. One is seed, which is your engineered cell population. Second one is fertilizer. Fertilizer by meaning where these reagents would support and survive your T cells, transplanted T cells, which are like antibodies, cytokine, and other cells. And soil is basically a preconditioned patients. So what they do is they take out blood cells from a cancer patient, which is called apheresis, and then they isolate out of these different leukocytes. They isolate very pure population of T cells, and then they expand with and co-culture with other cells like APCs, cytokine treatment. They do different treatments, and then they infuse. They expand into large population, and then they infuse after conditioning the patient. That means after slightly immune suppressing the patient. So the other category, the other way is to do is, if you look at the top panel, using a genetic tools to modify basically. So if I want to modify, so TCR I will modify, that is T cell receptor, I will modify to recognize one particular antigen, nothing else. So only one particular antigen I will modify, so that wherever that antigen is expressed, my T cell will go there and kill it. So these are licensed killers to go and do their job. Next slide. So this technology, because now we are going through this pandemic phase, this technology also can be used in viral therapies, viral treatment also. So we have some clinical studies going on with especially with HIV. Dr. Carl June's lab at Philadelphia, they have done this, some of the clinical studies in HIV patients. So the technology is same. Basically, they collect, because if you know the HIV pathology, it affects CD4 T cells basically, so that your immune response is knocked down, and then you have other complications. So what they did was they collected CD8 T cells, and then they inserted a CAR gene. chimeric antigen receptor, which I will be talking in my next slide. So they inserted a gene which specifically recognizes a epitope of that virus, which is probably GP160, and you can have several other antigens also. So they will insert that gene so that now this CD8 positive T cells will recognize only that particular antigen. So they expand into multiple numbers and then reinfuse. And what it does, wherever GP160 is found, it goes and kills that particular cell. Next slide. Now let us look into what are the technologies available, how they modified. There are two possible ways. One is natural way, that is TCR, T cell receptor. The other one is CAR, which is called a chimeric antigen receptor. So TCR is basically a receptor that has extracellular domain, transmembrane, and intracellular domain, which recognizes is a first signal for any activation, right? So they isolated that particular genes and modified them with the specific antigenic epitope. With nowadays there are many tools available, right, like transposons or retroviral vectors, lentiviral vectors, and most recently CRISPR technology, CRISPR-Cas technology. So you can specifically modify and induce that gene. So this particular T cell now is a modified TCR transduced T cell, which specifically recognizes only one particular antigen. On the other hand, you can have a chimera. Chimera means putting two parts together. So they have, if you look at the structure of antibody, Y shape, you have variable heavy chain and variable light chain for antigenic epitope determinant. So what they have done is they have cut off everything else. They just took VH and VL and then transduced into a chimera and then stitched into T cell. So which is called chimeric antigen receptor. So this also recognizes only one particular antigen and can go and kill that particular, wherever it sees that particular antigen. Next slide. So let us compare how the TCRs and CARs will fare. TCRs meaning T cell receptors and chimeric antigen receptors. So T cell receptor, if you see on the left panel, you have classical structure alpha and beta, and gamma, delta, and epsilon. Side by CD3 you have, that is on the extracellular side, you have transmembrane domain, and on the down of the alpha beta TCR receptor, you have zeta chain. This is a classical structure of CD3. And second, if you look at the panel B, so these are different CARs. So we are looking at first generation, second generation, and third generation. Now there is a fourth generation. So what is first generation? First generation means they just had a VH and VL. VH and VL fragments joined by a linker and a hinge region. And down they had a zeta, just like your TCR, but they did not have any CD3. So that is why it is called chimera. Now, this is simple CD3 structure per se. It mimics your CD3. Next, in the second generation, what they added was, they added a co-stimulatory molecule, which I described earlier, either 4-1BB or CD28. So it's a go signal. Once it identifies any target, it is a go signal downstream through zeta. Now, third generation, they had, instead of one go signal, they have two go signals, CD28 and 4-1BB. In the fourth generation, so what they had, which I do not have in this cartoon, now they have this third generation plus a cytokine producing transgenics. That means they induce either IL-2 or IL-7 or IL-15, which promote the proliferation of that particular T cells. Next slide. So if you look at the T cells, when they differentiate progressively, what phenotypic and functional changes happen actually? So in the down, if you look at the cartoon, where the tumor antigens are taken up by the APCs, which is basically a macrophage and a dendritic cell, and is presented to a available T cell. So if it presents to the naive T cell, for example, so it turns into T stem cell memory phenotype. And once it exposes to an antigen, it turns into T central memory, and further exposures will lead to T effector memory and T effector cells, and it gets exhausted. So in T cell immunology, you know that once you activate T cell, there is a concept called activation induced cell death. So once they activate, they perform their job and they die. So you can see that functional differentiation as it progresses, anti-tumor efficacy and self-renewal decreases, as I as I mentioned in the triangles here. And cytotoxic potential will be very high with the effector cells because they are bound to do their job. But can they live longer? No. Now, proliferative capacity, survival, and responsiveness for long term, if you see, so your ideal candidate would be T stem cell memory. So now people are looking at how to activate or how to engage a T cell with a specific tumor antigen and still keep it young. That means a stem cell phenotype. That is most of the people now research is looking at. Next. So if you look at genetically modified T cells, so what strategies are required to form effectively engineered genetic T cells which can do their job? One is target identification. You can do either TCR by or CAR, whatever is fine. With the advent of genetic technology now, I think CAR technology has taken over now, I would say. Then improve their function. Like how do we do that? Amplify the signaling domains or manipulate the hinge regions or prevention of TCR mispairing and dual specificity. This is also CAR dual specificity. This is also one of the important factors where you can have two chimeras that can recognize two different epitopes on the same cell, so that they will have a two different capabilities to recognize two different epitopes, but still can kill that particular cell. And then next part is proliferation survival. How do you make them happy and then proliferate? So that is by using different cytokines, proliferative cytokines like IL-2, IL-7, IL-15, or inhibiting some of the inhibitory signals like CD95, which is a death ligand. And then you have to also overcome the immune suppression, because when you train your T cells to do a killing job or to give a license to kill a tumor also will have response. So this will throw inhibitory signals at you. So we have to reduce those inhibitory signals also. Other option would be you can have a trafficking and engraftment also. If it is a solid tumor, you can have a trafficking with the specific receptors and then have a engagement also. And other alternative would be combination therapies, like earlier Dr. Honjo and James Allison, who discovered checkpoint blockade. So you can have a checkpoint blockade along with your trained T cells to do the job. Next slide, please. So if you look at this slide, we have mostly TCR clinical studies listed here. So it has been now increasing that more and more number of adaptive T cell therapy transfers for different kinds of cancers are happening, depending on your targets. But one important thing here is what important major antigen you have to select depends on that particular tumor type. So specifically, you have to isolate that particular gene and transduce into a TCR. Next slide, please. Now, this slide is basically about CAR T cell technology that is approved very recently by US FDA. So the story began somewhere in 1989 when the first CAR has been incorporated into a T cell, and from then to came to approval, it took how many years? Almost 20 years. So 2017 first approval got, and that is for B-cell acute lymphoblastic leukemia, which was by Professor June at Philadelphia. So he treated, and this is approved for young and adults up to 25 years of age. So there is an interesting story here, because one small girl from New York, so she has been through all therapies possible for her beta cell acute lymphoblastic leukemia. So, but relapse was heavy. So as a last option, then that time, it was around 2010. That time she went to a clinical site where Dr. June was conducting clinical site. So she went and then she enrolled for this. She was the first patient, and then now it is to 2020, and she is living happily and healthy. So there are the tumor is almost the lymphoma is almost reduced, and likewise you have other you know tumors for which CAR T-cell therapies are approved, which is very recent, as you can see from the list. Next slide, please. So now T-cell antigens, using antigens to modify T-cells and then making them TCR transgenic or CAR transgenic, is everything is fine, right? Now there are a number of T-cells which cell to select, because if you look at T-cell, a naive T-cell in a different cytokine milieu will turn into a different cell type. For example, somewhere in 1980s or 83, I would say. So where they discovered Th1 and Th2. So from there, the differentiation started. Now we have almost nine to 10 different kinds of T helper subsets. So out of these, which one would be better? Now the race is on to identify a best T cell subset that can have a best activity and have a longer duration. Here we focused specifically on Th17 phenotype. So our focus was just to see, because Th17 cell lines are known to generate IL-17, which is a pro-inflammatory cytokine, and it is very well known that it is important in the inflammatory diseases. Then we thought that we will use these cells to utilize. We'll use these cells to utilize in the way to properly train them and utilize in the treatment strategies. Next slide, please. Pramod, can you go to next slide, please? So now what we did, so we set up an experiment where we had a female 1 CD8 positive T cell. So this mouse, once you collect spleen from this mouse and specifically activate with a specific peptide, it only proliferates CD8 positive T cells. So this mouse model we used, we collected those cells, activated these T cells with the peptide, and then used the cytokine milieu of IL-6, TGF-beta, IL-21, and IL-1 beta to generate into TC17 T cells. So what is TC17? It is just like a TH17. TH17 is T helper type 17. TC17 is cytotoxic T cell which produces IL-17. So it is a CD8 positive T cell. And then we also modified, after differentiation, we also modified to have a dual role, where we wanted to check what if we add IL-12 and IL-23 to the differentiation culture, so that we will have different populations, or a particular cell can have two different efficacies producing TC1 and TC17 type phenotypes. So then we expanded these cells. Then we planned an animal experiment where we inoculated C56 black 6 mice with B16F10 melanoma model, which is a very well established model. So which is a very fast growing tumor. So once we inject the tumor, it takes around 5 to 7 days to grow the tumor. Then on day 7, we infused with these targeted TC17 and TC1 T cells, mixed phenotype T cells, into these mice. So we had two groups here, TC0. That means there are only T cells which are expanded with cytokine like IL-2. So they have not been differentiated to any subtypes. And the other group we had was TC17s. And then we monitored the progression of tumor Next slide. So if you look at this slide, this data is basically the in vitro data, where once we programmed these cells to become TC17, and how the cytokines and the transcription factors expressed on these cells. So panel A shows you TC0 and TC17. On the y-axis, you have either nothing treated, that means no treatment. They were induced with either IL-2 or TC17 inducing cytokine milieu. Next one was IL-12, next one was IL-13, along with differentiation in the case of TC17. If you look at TC0 with no differentiation or no cytokine added, you have almost 80% of cells producing gamma interferon, which is known. And once you induce with IL-12 or IL-23 added, then the percentage is reduced to around 60%. Whereas TC17, if you look at, most of the cells are primed and programmed to produce maximum IL-17, which is evident from here. As you add IL-12 or IL-23, you have different scenarios now, where some of the population, at least out of this 82 population, 41, half of the population went to double positives, where they can produce both gamma interferon and IL-17A. But IL-23 did not do that. It only produced IL-17. If you go to panel B, again, gamma interferon versus granzyme B, this panel. So you have double positives increasing with IL-12 treatment. If panel C is basically expression of ROR gamma T, which is a lineage marker transcription factor for TC70 or TH70, and TBET is a transcription factor for TH1 type. So they express their phenotypes. And panel D shows just the ratio, how the expression of transcription factor occurs in two different populations or two different cell types, and what is the ratio. Next slide, please. So now, if you look at the tumor data now this graph shows the tumor data, where panel A, so in the earlier slide, I mentioned that your host should be conditioned host. What do I mean by that? Once you have a patient, that patient should be immune suppressed, slightly, not fully, but you have to mildly suppress their immune system. So here what we did was, we gave radiation, because radiation is one of the therapies used in cancer treatment. So we used around 5-gray radiation in panel A, where we infused, if you look at, there is no treatment and TC17 only. So only these two cell types, and the tumor grew actually. Of course, TC17 has some significance delay compared to no treatment, but they grew. Now, panel A, second data, if you see, so these are IL-23 primed cells. So IL-23 primed cells, at the day progressed, it also delayed the tumor growth. But interestingly, when you look at the last panel, IL-12 primed TC17, then you have the delay is very heavy or very long, and then you have very significant difference between your control groups versus your treated groups. So if you can see that on y-axis, the tumor tends to grow as time progresses, and there was a regression with the TC17 IL-12 primed TC17 treatment. And also, if you look at female 1 versus host cells, so these cells were also there in the memory, because as we know that most of us will get vaccinated within 2 years of age. The reason is you have a memory, right? So because you will not have thymus, which is convoluted after you are adolescent. So what happens is that memory stays. So that's why we wanted to check whether this memory also exists in the hair. So you can see that with TC17, you have more number of cells with CD8 and CD4s and NK cells also, compared to other treatments. Here we had no irradiation, 5-gray and 9-gray. So 9-day is actually myeloablative, which means it completely knocks down your immune system, which would be very difficult to recover. So in that case, we have to give slight stem cell protection also. So that's why you have to infuse hematopoietic stem cells so that they survive and then see the activity of your infused T cells. Next slide, please. So if you look at this slide, so now we know that in ex vivo, when we primed our TC17 mediated T cells with IL-12, it had some improvement. Now we wanted to see whether I can have IL-12 primed TC17 cells as a treatment option, as therapeutic option. Once after I induce tumor and then use these cells as therapeutic agent, can I add IL-12 exogenously? So that came to our thought. Can we use IL-12 and can it be of any use? So what we did was we injected the tumor and then day 7 we started the treatments with different T cells. One is IL-12 primed T cells, primed TC17, and the other is IL-12 primed TC17 with either IL-12 or IL-23 exogenously added, which we gave like 0.3 microgram per kg body weight of the cytokine. That is subcutaneous we gave. So if you look at with no treatment, obviously as expected, the tumor grew, and exogenous IL-12 alone did not improve anything. It was as good as your control. And I'm just talking about panel A. So if you look at there, IL-12 primed TC17 cells have not shown much improvement after day 30, and as well as IL-23 primed TC17. But once you add IL-12 primed TC17 plus exogenous IL-12, then it showed durable responses, and we followed it up. Just wanted to see whether this is significant and how long it is significant, because relapse is one of the important factor in cancer therapies, any cancer therapies. So relapse is one of the important factors. So we wanted to see whether it will relapse again. So beyond 60 days, it slowly started to rise. So we followed it up to 90 days, but we stopped around 75 days or so. So there was a significant difference between IL-12 primed TC17 with exogenous IL-12. Now panel B, if you look at, now we wanted to look at, okay, now we have given, here we have not given any irradiation. That means we have not preconditioned the host per se. Now we wanted to precondition the host. That means our tumor containing mice. So where we wanted to see effect of radiation, what effect it has. So what we had was 0 gray with no irradiation and 9 gray myeloablative irradiation with HSC protection as well. There it showed TC17 was very potent in reducing the tumor, and there were almost no traces of the tumor at day 60. Because it started reducing around day 30, but it touched the bottom by day 60. There was nothing, no trace of it. And the last panel, if you see, these are TC zeros, means we have not differentiated into any other T cell type, but just improved the numbers just with the addition of IL-2. But the tumor tend to grow by 30 days because we could not follow longer because most of the animals died by then. So now what it says is the 3 important things. One is you have to precondition your host, use the right T cell, and then transduce with that right epitope and train it correctly ex vivo, so that when you infuse into the patient, they go and do their job. Next slide, please. Now, with any upcoming technology, there will be obviously hurdles, opportunities. Both are there, right? So we have to consider mindful. We have to be mindful about the harmful effects of this also. So what would be the hurdles? Because this is a highly personalized treatment, because you have to collect the T cells from the patient, expand them. That means training and expansion happens in laboratory, and then again you infuse them inside. So and which does not fit any of current oncological practices. So that's why FDA took some time to decide where we have to put it, because it is not a drug. So we can consider rather than a drug, it is a service. So now FDA in 2017 and 18, FDA came up, US FDA came up that so it should go as a immunotherapy. So that is a separate area available now for different cancers. So this is one of the treatment options. Now other issues are logistic issues because you have to have autologous blood products, autologous T cells, autologous everything, and you have to have large scale manufacturing facility where once you collect from the patient, you have to expand them and train them in the laboratory and then reinfuse them. So those facilities are, you require a larger scale, and you have to validate the process also. After I expand, uh, you have to have a quality control also. How good my expanded T cells are, because you know that once you expand, you have activation induced cell death also. So that angle also is very important. How I do a quality control on my expanded T cells and then make them ready for the infusion. Now, what are the opportunities here? We can have one way out would be a blood bank, because you will have cells and then stem cells you can isolate using CD3 positive T cells, and then you can train them. Once it is a stem cell, you can push them whatever you want and then train them what you want them to become. In that case, you can have allogenic cells also instead of autologous. That means you can take from the other subjects, but only thing we have to be mindful is HLA typing, because you should not have a mismatch. There is a mismatch, then is a rejection problem, graft rejection problem. Now, there should be also ideal location for generation for anti-tumor T cells for ACT, which requires not only basic scientists, because you require basic science, genetic scientists, and clinician scientists, which play a very important role here, because they are the ones which who knows the patient condition, control when to infuse, when to take out. So it is a very finely amalgamated thing. We have to work together 3-4 different areas and come up with a therapeutic outcomes. And the topic, thank you for your patience, and if there are any questions, I would like to take them.
Dr. Sreenath sir? Yeah. Hello?
Yes, sir.
Yeah, thank you so much. It was an extensive view on the possible cell immunotherapy for treating various cancers, and your view and your experiences with the experimental model with the mice has been shown the CAR therapy as well as the T cell dependent cell mediated immune therapy and the possibility of the recombinant or the genetically manipulated cells for organ regeneration. So all these information have provided an extensive view on the possibility of cell immunotherapy or the cell therapy in treating various diseases and disorders. Now, I request Dr. Shankar to moderate the question and answer sessions. There are many questions that has been raised by the participants in the Zoom as well as in Facebook and YouTube. So we are consolidating them. A few questions will be asked to you to answer them because there are plenty and we don't have time to address all of them. So I excuse that from the audience sides. So for the participants. So now I request Dr. Shankar to monitor or moderate the question and answer session. Shankar, over to you.
Thank you, Dr. Pramod. Dr. Sreenath, it's an excellent talk giving an overview on cancer immunotherapy. There are several questions which are raised by the participants. We have listed out few of them. So,
yeah, I expect several questions.
Okay. How immune cells therapy works for human specimen? Is it through B cells or T cells or natural killer cells?
So far, it is based on T cells. There are NK cells also now in the trials. Now, approvals, whatever US FDA approved and other sites throughout the world approved is basically a T cell mediated immune therapy.
Fine. Okay. Okay. The next one will be how engineered cells escape immune rejection.
That's a good question, because these cells are trained to recognize their particular only single type of antigen, and if you have a dual specificity, they will recognize 2 antigens. Now, what happens is, that is why if you look at the cars, fourth generation, what they are doing is they are doing additional thing where, because if you see all the first, except from first generation, second and third generation had their stimulatory molecules already had added into the before the zeta chain. If you look at the intracellular fragment, either CD8 or 41BB or combination, so that it has an intrinsic signal, that is signal 2 already intrinsically present. So it doesn't care about whatever inhibition that is getting from the tumor.
Okay, thank you. The next one will be, is there any technique that can increase the particular type of immunity?
I'm not too sure about what we meant by this, because if you want to push a particular population, you can do by cytokine milieu, you can push a population. But in general, I'm not too sure what the question meant.
Okay, thank you. So this question will be relevant to the current scenario. How immunity against the genetic engineered organism concern to corona and biological warfare?
Yeah, this is a very relevant question. So now with the corona or whatever COVID-19 we are seeing, the establishment of a particular major antigen would be one of the limiting factors right now. That could happen because once you have a virus changing its epitope, it would be very difficult to predict, because vaccination also, you need a stable epitope, right? So you need a stable epitope to recognize by the immune system and attack. If it is changing frequently, then it would be difficult. In such cases, what we can expect is targeting a mRNA directly. That means either RNA vaccinations, that is now technology available. So I think as I read recently, Moderna has finished already a second phase clinical trial in the US, and they are going to do multiple site trial where they target RNA-based vaccines instead of protein-based vaccines. So that could be one option. And biological warfare, once you modify anything, unless you have an antidote, it is very difficult. You have to have a sequencing, and then you can have an antidote, and then whether it is a point block antibody or anything, so you can block it.
Okay, thank you. The next one will be, what is the role of cancer immunotherapy in patients with pre-existing autoimmune disorders?
That's an excellent question. So that is very good question. So yes, these cells are going like gangbusters basically, because they are licensed to kill, right? So wherever the antigen is there, it is going to go and kill. So that is why I mentioned that a clinician scientist is very, very important here, because you can control when to infuse. And the other option here would be, you can induce a TK gene, which is a thymidine kinase gene, so that they go after a certain period into cell death mode, so that they will not harm the other cells. That would be the other option.
Okay, thank you, Sreenath. I think I will take one more final question. Can it possible to design engineered cell vaccines which is a non-virulent to boost a specific immunity?
Can you repeat the question? I could not.
Can it possible to design engineered cell vaccines which is non-virulent to boost a specific immunity?
I would not say it is as vaccine, because I would say still as it is ACT, adaptive cell transfer therapy, which some of the clinical trials, at least we had a encouraging results from HIV trials, where they have used GP160, targeting GP160 and targeting your immune therapy using T cells. But vaccine per se, whole cell cannot be, because it has to be.
Okay, Sreenath, thank you very much for your clarification, and it was a wonderful talk. We'll look forward for in future to interact with you in future for your research updates. Thank you very much, Dr. Sreenath.
Thank you, Dr. Shankar, and thank you, Dr. Pramod and the organization committee, for giving me an opportunity to present my work. Thank you.
Yeah. Dr. Srinath, thank you so much. So on behalf of Department of Biochemistry, Davangere University, and the Society of Biological Chemists, Davangere chapter, so we heartily thankful to you for sparing your valuable time and discussing extensively on your topic of possible cell immunotherapy. So with this, we are going to conclude your sessions, and thank you so much. Thanks for being with us.
Thank you, Dr. Pramod. Appreciate it.
So very good morning. Sorry, very good afternoon. And sorry for some inconvenience with the network connect. So we are delayed for about 2 to 5 minutes. And with this, now I welcome you all for the valedictory session of the National Webinar Series on immunity and infection biochemical approaches for therapies. So in this national webinar series, which is organized from 15th June 2020 to 21st June 2020, there were 2 sessions each day. And the first session was scheduled at 11 to 12, and the second session was 12 to 1. So we run almost like a week with a lot of sessions which are covering different biochemical approaches that are possible for therapies targeting infection and immunity. And with this, I welcome you all to the valedictory function of National Webinar Series. Myself, Dr. Pramod Desan, I would like to extend a warm welcome to the valedictory function of the 7-day National Webinar Series 2020 that was organized by the Department of Biochemistry, Davangere University, and Society of Biological Chemists, India, Davangere branch. The present National Webinar Series was designed with the focus of creating awareness and educate the participants about immunity and infection. As we are passing with the wave of COVID-19 pandemic all across the world, the curiosity to understand the viral infection and immunity had reached all sectors of life. The distinguished speakers who gave a lead talk in the assigned session have delivered the message of managing infection and boosting immunity. I hope the topics of the presentations were very appropriate with the present global crisis, and it provides extensive knowledge on COVID-19 severity of disease, mortality rates, immunity, immune boosting, recovery chances, and possible diet and drug therapies, and so on. I hope the present planned seminar with these talks from distinguished speakers across the disciplines of biochemistry had made us understand the biochemical approaches for therapies to boost immunity and to tolerate infection. With these few words, now I take the opportunity to formally welcome you all to the valedictory session of National Webinar Series 2020. First, I would like to welcome our Honorable Vice Chancellor in his absence. I also take this opportunity to extend warm welcome to our esteemed Registrar, Dr. Basavaraj Bonakar, sir, for agreeing to deliver valedictory session remarks and to grace the occasion online. His support and encouragement was immense, and he is in driving force in organizing this event. Now, I extend hearty welcome to our convener of the webinar series, Professor Gopal Advirao, who is also the finance officer of Davangere University, for this occasion. I also extend a warm welcome to our organizing secretary, Professor Wadlapudi Kumar sir, Chairman, Department of Biochemistry, and Dean Faculty of Science, Davangere University, for being with us to give concluding remarks of the webinar series on this valedictory session. The delivery device of the sessions during this webinar series are our speakers. I thank all of them for agreeing to spend their valuable time during the sessions and nurturing us with vast knowledge of immunity and infection. I welcome all of them in their absence. The most important is the participation and enthusiasm from the receiving science lovers and audience who are the key for the success of this kind of webinars. I, on behalf of organizing committee and on behalf of Davangere University, welcome you all to this valedictory session. Finally, I welcome all the faculties, staff, and students of Department of Biochemistry. I welcome one and all who are present online to witness the session, valedictory session of this National Webinar Series 2020. Thank you. Now it's time. Actually, the valedictory session was supposed to be addressed by our esteemed Register Dr. Basavaraj Banakar sir, and I'm extremely sorry to announce that because of preoccupied schedule, he was not able to address the valedictory session. So, with an seeking apology to that, now I request our organizing secretary, Dr. Wadlapudi Kumar sir, to give a concluding remarks and an overview about the national webinar series on immunity and infection, biochemical approaches for therapies. Kumar sir, please.
Thank you, Dr. Pramod. Hope I am audible, right?
Yes, sir.
Yeah. I welcome all the participants for this valedictory session of National Webinar Series on Immunity and Infection, Biochemical Approaches for Therapies. It is a very good learning experience for us amidst this COVID-19 pandemic. So Society of Biological Chemists India, Davangere branch was started in our department in 2001. Since then, we have been organizing many seminars and conferences for the benefit of our students and as well as our faculty members. And this is the first of it in its kind. Through online mode, we could able to organize this particular webinar series for 7 days, inviting 12 academicians and as well as researchers from academia and as well as research institutions and corporate sector. And it has been very good experience for us how to operate the things through online mode, utilizing Zoom platform and as well as linking the same to Facebook and YouTube live stream. So this webinar series was inaugurated by Honorable Vice Chancellor of our Davangere University, Professor Sharanappa Vaijanatha Halse, on 15th June by 11 a.m. And he has given his inaugural remarks as president of the function. And similarly, Dr. Basavaraj Banakara, Registrar, Administration, Davangere University, who has been immense support for us. And he also proposed his inaugural remarks, and followed by Dr. Anita HS, Registrar Evaluation, Davangere University, and my own colleague in the department, Professor Gopal Madhurao, and Finance Officer in charge of Davangere University. He was also there to witness inaugural event of this webinar series. And I take this opportunity to give appreciations for my colleagues in the department, Dr. Pramod, Associate Professor, Dr. Santosh Kumar, Assistant Professor, Dr. Purnima, Assistant Professor, Ms. Chaya and Ms. Gautami, teaching assistants, and as well as my research student, Mr. Pratap, who is well versed with this online events, operating the things, giving the link to YouTube channel, and as well as Facebook, and Zoom operations. So the sessions were begun on 15th with the first talk by Dr. Santosh Kannade from Department of Plant Sciences, University of Hyderabad, who could able to deliver wonderful lecture on phytochemicals mediated epigenetic modulations in immunity. Most of the people have been working on phytochemicals and characterizing them for antioxidant property and as well as anti-inflammatory property, anti-cancer, so on and so forth. But the thing is how these phytochemicals, they modulate the things at molecular level by changing the gene expression pattern. So it is the one which is coming up nowadays in recent times. So that's what he could able to orient every one of us and enlighten with his own experience in his lab, and as well as some of the information available on the published literature. And second, it was only one session on the day of inaugural event. On 16th, there were two sessions, as Dr. Pramod already highlighted, and you all people, you have witnessed this. It was by BC Girish from Veterinary College, Hassan. So non-clinical safety evaluations for drugs and biologicals. So he's a veterinary doctor who could able to throw some light on how the non-clinical safety evaluations can be done as far as drugs are concerned. And followed by the second session on repurposing drugs, hydroxychloroquine and as well as azithromycin being suggested for COVID-19 coronavirus infection. So how these drugs, they can be useful along with zinc supplementation to tackle with the viral infections. That's what Dr. Rajesh could able to enlighten us. And day 3, it was on 17th. This first session by Dr. Prabhakar BT from Kuvempu University. He could able to share his research experiences which were carried out in his lab and as well as from other reputed laboratories across the world. Slacking the neovasal sprout through natural products, the same thing which Dr. Santosh Kannade has given a different direction of epigenetic modulations, and Professor Prabhakar could able to show how natural products can mitigate or can be suppressant for neovasal formation as far as cancers are concerned. And the same day, there was second session by Siddharazu, understanding autoimmune arthritis and osteoimmune crosstalk. And fourth day, it was begun on 18th with Dr. Puneet Kumar from Alzheimer's, Mysuru, genetic testing in immune disorders, followed by Dr. Purnima Priyadarshini on food bioactive as immunomodulators during viral infection. Dr. Purnima Priyadarshini from CFTRI, Mysuru. So these were the people who could able to show how the food and nutrition and as well as the natural products, they can be useful to tackle this particular kind of epidemics and as well as pandemics, not only this, even certain cancers also. And it has been a very good exposure for our students. I think the same with you for almost all the participants also. And these events have been witnessed by on Zoom. We could take the permission for only 100 participants because I had an idea to link the same to Facebook and as well as YouTube channel to attract more number of participants. I thank all the participants who have all proved that it has been almost more than 500 every day on an average through all these 3 modes. And on 19th June, the first session was by Dr. Vishwanath Chachadi from Karnataka University, Dharwad. He could able to throw some light on MSC molecules as central regulators of adaptive immunity, as adaptive immunity plays a major role as far as these kind of epidemics and pandemics are concerned. So he has given some information about. And followed by Praveen Kumar, the experimental methods in immunology, the basic immunological techniques and as well as advanced techniques like facts and all. He could able to enlighten us with his knowledge. And day 6, it was Dr. Harish Prashant, how polysaccharides can play a role in immunity and as well as immune responses. That's what he could able to highlight the information about, providing the information on role of carbohydrates, especially polysaccharides. as we all know that polysaccharides, they may not be able to provide the long-lasting immunity, but still that are very important as far as the immunity and immune responses are concerned. That's what Dr. Harish Prashant could able to enlighten us. And followed by the second session was to be addressed by Dr. Divya Prakash from University of Pune. Sorry to convey to everyone, due to sudden demise of her father, she could not able to turn up. So that's why uh, we could able to make some alternative arrangement by inviting Dr. Nagaraju from Tumkur University Department of Biochemistry, who could able to enlighten us with experimental animal models on in biochemistry research. So what are the different kinds of animals that are used for the experimentation, and how different alternatives can be chosen? That's what he could able to show us with his expertise. And today, this is the last day, 7th day, the first session. It is by Dr. Sreenath Kundami, Head Cell and Molecular Biology, Laila Nutraceutical R&D Center, Vijayawada, Andhra Pradesh. Just now, we all witnessed cell-based therapeutics, focus on cancer immunotherapy. So these are the events which we could able to organize. Actually, we planned this webinar series for the benefit of our students, and our faculty members, they expressed that why can't we go in a big way? So that's what we named it as national level webinar series, because there have been resource persons from 4 states. Of course, we could miss one from Dr. Divya from Maharashtra, from University of Pune, Dr. Divya Prakash. So they have given the acceptance from 4 states, from Telangana, Andhra Pradesh, Karnataka, and as well as Maharashtra. So though it is our first experience, first time we are able to venture this particular kind of event through online. So with your participation, it has been made a successful event, and we thank everyone who supported us, and as well as I mean directly and indirectly, directly by participation and indirectly from encouraging us for taking up this particular event. And it is the brainchild, actually this event is brainchild of Dr. Pramod, in fact, and supported by Dr. Santosh and we all people. It has been a very good teamwork as far as our efforts are concerned. And sure, definitely we will, with your support, definitely we will take up many more events of this kind for the benefit of not only our students and faculty members, but also our peer groups from different institutions and different organizations across the country. So with these remarks, I'd like to appreciate the efforts of my colleagues, and I'd like to thank everyone who supported us once again. Thank you very much. Now it is over to Dr. Pramod.
Thank you, sir. Thank you, sir. Thank you so much for giving an extensive view and the overall of this webinar series that is organized, and about the speakers and the importance of the theme, and for the concluding remarks on the national webinar series. Now it's time to listen to the vote of thanks, and I request our faculty member, Dr. Santosh Kumar, to address the gathering and to deliver the vote of thanks. Santosh sir, over to you, please.
Thank you, Pramod sir. I hope I am audible.
Yeah, yes, sir.
Okay.
You can go ahead.
Okay, sir. Okay. Respected dignitaries, speakers, resource persons, dear participants, our colleagues, and my dear students. Good afternoon, everyone, and namaste to all. I am Dr. Santosh Kumar, Assistant Professor, Department of Biochemistry, Davangere University, Davangere. I consider this occasion as an honor, privilege to extend vote of thanks on my own behalf and on behalf of Department of Biochemistry, Davangere University, Davangere. First of all, I would like to thank our Honorable Vice Chancellor, Professor Sreenath B. Halse sir, and also express my honest thanks to Professor Basuraj Banakar sir, Registrar Professor Anita H.S., Registrar Evaluation, Davangere University, Davangere, for their encouragement and constant support for successful completion of this one-week webinar series, Immunity and Infection Biochemical Approaches for Therapies, from 15th to 21st.
Hello, Santosh sir, can you hear me? Yes, Santosh sir.
By Department
Yeah.
of Biochemistry, Davangere University, Davangere, in association with Society of Biological Chemists, Davangere branch. My heartfelt thanks to our convener of this webinar series, Professor Gopal M. Advirao sir, for his constant support and encouragement. My thanks to our organizing secretary, chairman, Department of Biochemistry, Professor Wadlakudi Kumar sir, for continuous support, encouragement for conducting this type of webinar series in our department. On behalf of Biochemistry Department and Society branch and my wholeheartedly extend my heartful thanks to each and everyone, all the speakers, resource persons of this webinar series from different organizations, different universities, different research institutes for their sharing their knowledge with their busy schedule. In spite of their busy schedules, a big thanks to all speakers of a important for this webinar series, Dr. Santosh, University of Hyderabad, Dr. B.C. Girish, Veterinary College, Asan, Dr. Rajesh, University of Mysore, Dr. Prabhakar, BT, Kuyampu University, Siddharaju MN, Dr. Siddharaju MN, University College of Mangalore, Punit Kumar, Director, Azymes Limited, Life Science Limited, Mysore, Dr. Purnima Priyadarshini, Scientist, CFTR, Mysore, Dr. Vishwanath Chechdi, Dr. Praveen Kumar SK, Karnataka University, Dharwad, Dr. Arish Prashant, Scientist, CFTR, Mysore, Dr. Nagaraj, Tumkur University, Dr. Srikanth, Head, Cell and Molecular Biology, Laila Nutraceuticals R&D Center, Vijayawada, Andhra Pradesh. I would like to thank our dynamic faculty members of our